Okay, so thank you very much for joining us today. I think Chris laid out quite nicely what we're going to cover today. We really wanted to try this first presentation, which is based on a lot of feedback that we've been gaining from lead investors and various others about how we present the business moving forward as we start transitioning to positive EBITDA this year and getting to scale. I think the story becomes a little easier to tell, particularly as we've done some of the consolidations that were necessary to get the footprint right for the business moving forward. So hopefully this will be a good road test. We'll also be finishing on some of the KPIs that we sort of plan on using, and getting some of your feedback at the end of this would be great on that as well. Okay, so if we start with how we position ourselves, we always position ourselves as Illumina is to DNA, as Abcam is to proteins, Horizon is to cells, because cells are the ultimate point of functional dysfunction for disease, for human healthcare. So if you're in a position to be the cell builders, you're in a position to design, engineer, and apply cells for the advancement of human health. Whether that is a product, a service, or the cell is actually a therapeutic or component in a diagnostic for a therapeutic is kind of irrelevant. It's all the same process of how you design, engineer, and apply that cell. So just to give you a bit about fundamentals, so the business is coming up to its 10th anniversary. It was a virtual business in 2007, seed funded in March 2008. It's built around gene editing, so it's a suite of tools that enable us to effectively design and engineer these cells at will. We're a picks and shovels business that enable people to develop a new class of medicines, so-called personalized medicines. These can be small molecules, antibodies, cells, or gene therapies, but they're all designed at modulating a disease phenotype in cells, whether it be at the genetic level, or the protein or cellular level. We're a growth stage company, a global company. You're on about a third of the company's footprint. It's about 32,000 square feet, and our footprint globally is around 85,000 square feet. Most of the in vitro activity is now consolidated onto this site, with a small site in Vienna, and all of the in vivo operations are based between Philadelphia and St. Louis in the United States. We're increasingly becoming a product business. So if you'd have seen at IPO, we had around 700 products in the inventory. That's expanded to 23,000 plus now. So we're increasingly sort of effectively productizing our IP. Export driven, over 90% of our revenues are exports. If you wanted to a rough sort of split, it's around 50% in the United States, 30% in Europe, 20% in the rest of the world. We have a reasonably small domestic market here in the UK, around 5%. So listed on AIM since March 2014. And clearly 4 of the analysts, I think 3 are in the room, are covering the stock. We're hoping to increase that throughout the year. So if we give a quick snapshot based on our trading update, and then we'll give you some more color on that. So we reported at least 24 million of revenue. That's 19% growth over the previous year. We expect EBITDA to be in line with expectations. A cash headroom of 11 million, and that should be sufficient headroom to get us through to positive EBITDA. Gross margin expansion from 49% to mid-50s, and we've given very confident revenue guidance for this coming year of 30 to 35 million. We have a very, very strong order book, over 50% visibility on that order book, so we're quite excited by that. And we committed at the IPO to become positive EBITDA this year, and that's what we're going to be achieving. So just a bit of step back. You're familiar with the sort of macro drivers of the pharmaceutical industry. Whilst increasingly investment has gone in and people have started this shift towards developing medicines that are more targeted to particular genetic drivers, pharmaceutical productivity is still in decline. It hasn't arrested the thought yet, because these medicines, whilst they are targeted with better, clearer reimbursement strategies based on companion diagnostics, ultimately you get a lot of resistance pretty quickly with these molecular therapeutics. So actually the productivity hasn't actually increased and translated into return on investment. So increasingly you're going to see a lot more of these drugs developed. Over 60% of oncology drugs, for example, that were filed for IND are based on so-called personalized medicines targeted to a particular molecular driver. And the objective is really to build a toolkit of drugs. Having a few individual drugs is fantastic. So when you have Lynparza for BRCA deficient, P10 deficient BRCA breast cancers, prostate cancer, these are great drugs, but resistance actually occurs quite quickly. And to have a real serious impact on disease like cancer, you're going to have to hit 2 or 3 different points in the weakness of the tumor to be able to achieve it. So until we get a critical mass of these medicines, it's going to be pretty hard to mix and match and have serious impact on disease. So there's a real demand there. If you look at the drivers behind personalized medicine, you're very familiar with the Moore's Law diagram. Sequencing is no longer the bottleneck, it's a commodity. We will understand all of the genetic and epigenetic drivers of diseases of aging like cancer, Alzheimer's, I believe, over the next 10 years. The issue is now, how do you translate that sequencing information into increased productivity? Well, the first thing is you need to develop better validated targets, so identifying targets that have a clear patient relevancy up front and develop medicines to those targets. And the key here is that you can develop drugs in a much shorter periods of time. So you've seen recently Tagrisso within 3 years, crizotinib within 7 years. Increasingly, drugs now are not taking 15 years to develop, but those drugs are targeted to much smaller patient populations. But if you have clear reimbursement strategies based around a companion diagnostic, you can get significant returns on investment as long as you're prospectively developing drugs against those patients and not retrospectively finding out that your patient population is much smaller than you thought at the end of the game. So the key is being prospective in this approach and developing critical mass. But these are some interesting stats where I think it's pretty sure this is going to happen. If you look between the first genome and 2015, only 1 million genomes across all species were sequenced. Over the next 5 years, if you look at BGI studies and etc., they're estimating 500 million genomes will be sequenced, human genomes, in the years 2015, 2020. So having all of the genetic questions are going to be there. The key is, how do you provide answers? And Horizon's business is very much positioned in the answers business. How do we provide people with the picks and shovels to create those associations between those genetic anomalies and disease outcomes? Now, we're talking about health here, but we aren't limited in gene editing to health. Think of the 3 major challenges of the aging population. How are we going to feed, fuel, and heal our population in light of statistics that show that age increase per day is 1 to 5 hours, whether you're in the developed or developing world. The first person has been born who will live to 150 statistically, but will they live well? And certainly as they get to the ages of 60, 70, 80, will they be economically and societally productive? Well, if you want to improve feeding, the principle is the same. If you understand the genetic trait of crops, you're then in a position to be able to edit those cells and increase the yield, improve the flavor, improve resistance to sort of pests for those crops, etc. Or if you want to actually fuel, you're in a position to understand the genome of cells that are going to metabolically produce energy. You're in a position then to improve that. So when you think of Horizon, there's 3 processes. It is understanding the genetic trait, linking it to an industrial or healthcare outcome, and then manipulating the cells to provide either picks and shovels to enable other people to enhance that process, or actually use the cells themselves to enhance that process. So when you think of Horizon's future, health is where we've been historically focused, but we're increasingly now working in all types of gene editing, and you'll see lots of collaborations moving forward in different areas. Okay. So in summary, the business has been built very much on historically cancer, because cancer is very much a genetically based disease, but we are not a cancer technology. We are now supplying products and services into all different therapeutic areas and operating there, but the principle is the same. There's a genetic feature that is the difference between health and disease, and we're able to do 2 things. We're able to recapitulate that genetic situation of health and disease in a test tube or in an animal model, for example, and they're used at different stages of drug discovery and development, or we're actually in a position to be able to edit the cells themselves, for example in autologous T-cell therapy, to be able to then correct diseases in patients. Chris is going to give you the detail. I'll give you the Rod, Jane and Freddie at the top. So gene editing you're all familiar with. It's making very specific changes to a gene. These can be insertions, deletions, substitutions, rearrangements, amplifications. Pretty much any change that you would want to do to a cellular genome, we're able to do that. And the reason we're able to do that is 2 reasons. One is a decade of experience. It's still not easy when you want to do really complex genetic modifications. You also want to contextualize those modifications. You may want it to be inducible, conditional, lots of different ways you want that sort of genetic variation to present itself. And we're experts at doing this gene editing. We have access to multiple technologies. We built the business on the viral technologies, RAAV, which Chris will tell you is very, very precise. It's a little bit more laborious, as you have to do 1 edit at a time per allele, but it does it with absolute precision. So I think of that as a scalpel, if you like, of a surgeon's toolkit. If we move to the zinc finger nucleases, These can be engineered to be quite precise, so they're scissors for specific effects. So they're very sharpened scissors, they're not the general draw scissors. It's quite expensive to make, but they're very good at doing things like animal editing in mice, rats, and bunnies, and we have it exclusively for that in mice, rats, and bunnies. Then you move on to the CRISPR, which we think is the Swiss Army knife. It's flexible, it's easy to use, you can pull it out, do lots of things, but you wouldn't necessarily want a non-expert performing surgery on you with that toolkit. And then there are improvements to that toolkit that we're working on. So we've got a novel transposase technology. It's the world's first cut and paste transposon, so copy and paste transposon. So think of it like the photocopier. Once you've got an edit you want to make into the genome, you want to repeat that edit through the cell very rapidly and precisely, and that's proprietary to Horizon. And there's some novel techniques that we're looking at now in other endonucleases called Argonauts, and we're looking at those to get a proprietary therapeutic position in that space. So we have all the tools in the box, and that means we're in a position to effectively make any type of edit that you want. And Chris will talk a little bit later on one slide about, without giving you too much detail, about our T-cell therapy platform and the combination of techniques like RAAV plus CRISPR, for example, overcome some of the fundamental limitations in certain areas of these techniques. So, top level business model. We believe the model we have is highly scalable and leverageable. So if you think of it, we use gene editing to develop engineered models of human disease, whether they be cell lines or animals, and it's every type of cell line that you could imagine: stem cells, somatic cells, T cells, all of those different types of cells. And we have effectively a catalogue of 23,000 cell line and related cell line products. So they may be a cell line that has a knockout, a knock-in, a reporter tag. They might have an inducible cassette in them. They may be a stem cell, a somatic cell, all different types of cells, and animals too. So we have lots of different therapeutic areas for neurological, cardiovascular, cancer, etc. Models, pairs of rats, pairs of mice, etc. that represent those genetic features. And those cell lines and related products are licensed for research use to customers. The cell lines are converted into valuable downstream products. So those cell lines can be very specialized cell lines that are used to manufacture pharmaceutical proteins to GMP. So a much higher value cell line than a research use cell line. They're actually being used by pharma companies to manufacture their drugs. Or the cell line is being derived in many different ways to produce reference standards for genomic processes, whether they be research or clinical. All formats, next-gen sequencing, for example, FISH, IHC, different formats, DNA, RNA, protein, etc. And they're all derivatives from the cell. Or the cell lines are used in a range of contract services, so everything from target identification right the way through drug discovery development, through to preclinical services and drug combinations, actually matching the drugs that are going to work best for patients. But the bottom line is the same cell that is the basis for all of these offerings. So we consider these cells like assets that can be leveraged into multiple revenue streams. So looking a bit at markets, you've seen our annual reports, the global macro markets in these four main areas: drug discovery, development, manufacturing. So these areas here, basic research, drug discovery, manufacturing, molecular diagnostics, are over 29 billion, but that's kind of a meaningless number. So we've actually really sort of do more of a bottom-up analysis and looking what our served available market is and where we sit in line with that. And if you look at these areas here, 300 million for basic research, 600 for drug discovery, 200 drug manufacture, and 500 million for molecular diagnostics, growing at really nice sort of rates. So that totals about 1.6 billion, which we think is our served available market right now, and we have around 2% of that market share, and our plan for the next number of years is how we get that to sort of 5% market share profitably. One thing to note is the gene editing component. If you looked at the market assessments for gene editing just a few years ago, they've been already outstripped. So the 5-year forecast have been outstripped within 2 years. And that will continue to be the case. What's happening is there's a displacement of a lot of these activities with gene editing related substitutes. So when you look at cell-based assays market, which macro is over a billion, right, the proportion of that was historical, just cells from tissue banks, is being displaced now with edited cells with the availability of CRISPR. So this is really attractive for us because most of the competition consists of substitutes that are in the process of being replaced. Now the technology is available. So we're not having to create fundamentally new markets in every area. So I think this is the slide that I'd like to sort of really take as a take home, because this is the business in a nutshell for the next 5 years. If you think of the 3 years since IPO, we said we would move very quickly to scale the business, fill out the technology platform from sequence to treatment, and we would, over a period of time, balance the business so that the products business would first become 50-50. When we came to public markets, we were 85% or more a service business. We're now around 55-45, and we expect that to get closer, and over time, over the next couple of years, the products business to far exceed the services business. So when you think about business over the next 5 years, this is our strategy. So our product strategy is very much about how do we deliver a 30-40% revenue CAGR at a greater than 70% gross margin. If you look at 2016, products was at least 44%, at least 70% gross margin. So we want to maintain those high CAGRs in the products business and increase that product margin from 70% upwards. So we think we're in good shape. We already did that in 2016, and we want to continue to do that over the next period of time. The services business, we want to rationalize a little bit, because the services business for us is not about how do we build a mini Charles River, mini Quintiles, etc. It's the shop window. How do we use this to do the very novel applications that our customers use the technology for? So effectively it adds value to the products, it adds data to the products, and it builds our knowledge base of this cutting area of personalized medicine, including cell therapy. And that means we're in a position to also leverage that infrastructure to get some of the upside in research biotech. So we're looking at a 15-20% revenue CAGR over that 5-year period at a greater than 50% gross margin. So if we look at the particular product profile that we're looking for, there are 3 components to it. There is a transactional layer off the shelf. So this, if your comparable is very much a company like an Abcam or Sigma Catalog, etc., where it's purely transactional. People are paying via the internet, they're paying people who are picking up the phones, calling them. You have less people in field chasing it down because that's expensive, and you get this predictable revenue. So you can't see it, but you know it's coming based on transactional history. The second piece, which I think is a much more valuable piece than that, actually, is what we call the OEM layer. So this is where you get visible, predictable revenue. So your product is becoming, it's much like ARM, the design inside someone else's product. And the 2 ways we do this are in both research and in clinical diagnostics. So a good example in research would be our relationship with Abcam, where we're just supplying validation of their antibodies using our knockout cell lines. So we can manufacture them at very high throughput, we can validate their antibodies. That gives their antibodies premium placement in the market, so we get paid for making the cell lines, paid for the validation, and we get a royalty on the antibody. And that relationship we hope to expand greatly as Abcam's business expands. The second area is on the diagnostic kit. We're in workflows for around, it's getting up towards 30 now, diagnostic workflows. We expect the first ones to actually start being approved in customers' kits over the next 12-18 months. So this is a product now where your product is used for the validation of a diagnostic assay. It could be next-gen sequencing for a company like Illumina, it could be real-time PCR for a Roche, or you've seen deals that we've announced over the time. You get paid for the validation materials for that test. They take the test through the clinic, you get paid for lots of materials for their clinical trial, and then if it gets approved, you're embedded in the kit product and getting a proportion of the revenue of that kit, typically 5-15%, depending on the volume, for the lifetime of that kit. So it's like a paid for product development, very high margin off the shelf for clinical trial work, and then when you're in the kit, it's all annuity on there. So the margin is much, much higher once you get onto an approved kit product. So for those OEM, you're typically getting a 50% or more margin for the development phases, but once you go in kit, your margins are really, really high, above 80%, even up to 90%. Really depends on the royalty rates that we have to pay for different licenses. And then the final bit is the mandated piece. So this comes back to the OTS. We have an FDA strategy. It's in its 3rd year now. We anticipate, I think we've said before, that we anticipate 2018, 2019 being where we're hoping to effectively get an approval from the FDA for suites of our products to be approved for use in diagnostics. On the services side, 2 things we do in service: paid for product development, so where people pay us to make animals, pay us to make cell lines. We do get to keep, not as many as we used to. It used to be 100% we got to keep, but it's because it's getting a bit more competitive, we do have to allow some products. So if it's something that's very bespoke to a customer that no one else is ever going to want to buy, there's no point retaining the rights to that. But where there is opportunity, so for example, all those diagnostic products, when we make the cell lines that get used for the diagnostic, we retain the rights to the cell lines to sell in drug discovery, as opposed to the diagnostic application. So you do get paid for product development. The margin is lower on gene engineering, particularly animal gene engineering, as you might imagine. You're not growing cells in test tubes, you're growing embryos in mice, for example. So it's greater than 40% margin. But the application layer, when we do things like genetic screening, molecular screening, assay development, that's greater than 60% margin. Okay, just a quick snapshot of each area. So that's, if you visualize it, that's what it looks like. Cell line catalog, animals, biomanufacturing cell lines, diagnostic reagents. That's the data from last year, 44% growth in that year, greater than 70% gross margin. The channels to market for this are inside sales, so it's people picking up phones, and we do have a small number of field-based sales representatives. E-commerce is increasingly becoming part of this. We'd like to drive that e-commerce adoption into the 20-30%, and then if we could get up to 50% in the long term, that would be fantastic. OEM partnerships, that's easy sale because you're just making revenue based on someone else's sale of a product, and then we use distributors in geographical locations that we're not prepared to go feet on ground. All I wanted to pick out on these slides really just to give you how these products are used. So if you think of the value to the customer of our research use cell lines, they enable customers to understand the genetic basis of disease. I have a genetic hypothesis from sequencing studies, and I want to functionally validate that hypothesis. So they buy a cell line from the inventory to do that. So it's basic research. Once you've validated a hypothesis, it's the same type of tools and derivatives that are used in drug discovery and development. So if you look at competition, the competition are traditional standard cell lines. Cancer cell lines, for example, where there are lots of genetic diversity in those cell lines, so it's not genetically defined. So you have to use a lot of them, it's very expensive, and so it's actually really not practical anymore to use those as competition. The new way of doing things is you hone in on the genetic feature of interest, and once you've developed a hypothesis and a drug against that hypothesis, then you go broader into the broader cancer mosaic, for example, to see how that would affect lots of different patients with lots of different genetic contexts. So it's actually a new type of offering. For research use catalog for animals, similar basic research drug discovery. For example, they're more advanced models. You have to go through these if you want to develop a drug that's going to go into a human, so people have to use a mice, rat, bunny model effectively to do that. Biomanufacturing, that's about drug and vaccine manufacturing. And the beauty of this is it's a very scalable part of the business. At IPO, I think, Mike, it was about 100 grand in there. Well, let's just say that's changed in terms of that. I think we've mentioned publicly before that we think this is a 10 million a year revenue line as you get to the end of this decade. You don't need lots of people to sell it. Once you've got a cell line, that cell line can be used by up to, there's 1,000 companies in that space, including 100 top pharma, so you're trying to sell throughout that different sort of customer layer, and it's a much higher value cell line, in the 150 to 350,000 range. And if people are paying you to make their own, you can actually get a milestone downstream in their drug value chain as well. Clinical use reagent catalog, so this is where people are buying products for existing tests that are already on the market. So these are clear labs who buy these products as research use only products to standardize their diagnostic workflows. Just to give you directionality on how the revenue is broken down, so the size of the box is the relative sort of proportion of revenue for each of these areas for the products. So that's the breakdown by segment, and down here at the bottom we give you sort of typical order size ranges. So for the academic customer, its products are down to from 500 quid up to 100,000 in terms of order sizes. If we look at the biotech customer, 500 to greater than 1 million in order sizes, and the same in pharma diagnostics, 500 quid to 1 million. So people are either buying pretty cheap cell lines from the inventory or specialist cell lines at a higher price, or specialist reagents, or they're buying bulk orders, etc., across those different areas. So you can see a lot of research use cell lines in academic. As you move into biotech, you start getting a lot more clinical applications, drug manufacturing applications, and the research use is smaller as a proportion of revenue in the pharma diagnostic biotech. A big focus for us moving forward is the academic customer base. How do we sort of get into that much broader layer of tens of thousands of customers? So that's a focus for us. Moving on to Horizon Services, there are three types of services that we offer. Disease model manufacturing, so modeling cell lines and animals, and they're deployed by customers in research. Contract research and development, so that's actually doing all types of services from target identification, validation, through drug discovery and early preclinical development, finishing with drug combination screening to find the right combinations of drugs targeting particular genetic drivers of disease. The third type of service we have is very much automated screening, and there are two types. There are genetic screening, which is very much based on identifying new targets or finding new genetic contexts for existing drugs, and molecular screening, whether that be single agent or combinations, to identify optimal therapies for patients or patients for clinical trial. So as we reported in the trading update, non-molecular screening services grew at 30% this year, but that was offset by our decision to consolidate the Boston site and reduce screening capacity in 2016 by 2.7 million. And we'll come on to that in the second part of the presentation about the benefit of that in saving 5 million of run rate costs in 2017. So overall the business grew at 4% there. The channels to market here are a bit different from products, so it's very much a field business development team supported by field application scientists and by scientific alliance managers that go out with the team to sell this high-level scientific research work. We're very much built on deep relationships with pharma, over 30 of the top 50 pharma, and driving repeat business with existing clients. So if we look at examples here in the custom cell models, we're very much focused on delivering models that enable people who have a genetic hypothesis and to link that to a disease outcome, whether it be a new drug target or a diagnostic target. And so from the basic research, we provide models to validate a hypothesis and then advanced models and tools to enable people to develop their drug and/or companion diagnostic. On the contract R&D, that's us actually deploying the picks and shovels to find gold on behalf of the customers, and customers come to us because we are experts at deploying our picks and shovels. In terms of genetic and molecular screening, as you'll see on the lab tour, this is about automating the capability to perform certain types of services in high throughput mode, and the ability to design engineer cells and related biological content enables us to scale up the work that we're able to do without adding extra headcount and thus cost. Again, looking at the revenue breakdown here, if we look at academic customers, very much focused around genomics and functional genomics, so the disease models are the best selling opportunities here. Typical order size is 5,000 to £500,000. We do get genetic and molecular screening and contract R&D from some of the bigger core facilities. Our biotech and pharma customer segmentations are pretty similar. We get a lot of contract R&D from target right the way through to clinical trials, preclinical work, and our genetic and molecular screening and our making of models to using those screens is about equal to. Typical price sizes 5,000 up to 1 million for biotech and up to 2 million for pharma diagnostic companies. Coming to our research biotech unit, which effectively is our R&D capability and leveraged in terms of its business model, two types of upside we develop here. One is the historical milestone and product royalty portfolios, which is based on a partnership model with big pharmaceutical companies. Here we're very dependent on what the pharmaceutical company and the partnership actually develops with those new drug targets or drug candidates, and we get milestones and royalties based on pathway through preclinical and clinical. We've increasingly moved over the last 2 years to a spin-out model, how we actually crystallize out a value for our research in a new equity-based venture. So 3 areas we're focusing on: immuno-oncology, which is redirecting the immune system to tackle cancer as though it's an invading organism; synthetic lethality, which is finding the genetic Achilles heels of cancer that are caused by actually those cancers losing the function or a mutation of a particular gene. It creates compensatory mechanisms that if you can identify those genetic contexts and take them out, you'll cause a diseased cell to die versus a healthy cell. And we've developed a whole range of target assets there, which we're in the process of putting into a spin-out venture and getting funded by third party. And then in gene and cell therapy, as you'd imagine, as the cell builders, we're well positioned to take cells from patients with novel targets, engineer those targets, and actually create those cells, if you like, as therapeutic weapons to actually cure diseases. So historically, we have spun out Avinity Therapeutics. We're a major minority shareholder in immuno-oncology, and that will be refinanced in 2018 by third parties. And we're in the process of formulating spin-outs for our synthetic lethality assets and our cell therapy platform. And again, what we're looking for moving forward with this part of the business is that we get a valuation of the net asset value of this portfolio of equities, milestones, and royalties. And as projects are refinanced, we get an increase in that net asset value, which can then be marked up as additional value beyond the products and services revenue. Customer base continues to increase. We're approaching 2,000 unique customers right now. So each customer here, whether it be academic, pharma, biotech, diagnostic companies, or general research companies, that's starting to increase. We sell into over 50 countries now. So moving forward, as well as being the cell builders, we're very much committed to being business builders and delivering what we said at the time of our IPO to a business that would be positive EBITDA in 2017. So the four ways that we do this is by continuing to address very high growth markets, delivering sustainable, repeatable business on a footprint that can be afforded to deliver profit, delivering our flywheel model. We have that priority for a scalable products business, which is transactional off the shelf, and OEM or business to business relationships, rebalancing the services to optimize revenue growth, margin, and profitability, delivering upside from the value of our core IP innovation into our value chain. Focusing on what we do at best as the cell builders, building our business around two centers of excellence, in the UK, the in vitro offering, and in the US, the in vivo offering, and scaling our business through operational and commercial excellence. And the bottom line is we are going to deliver what we said we're going to deliver. Delivering that top line growth and upside, commitment to positive EBITDA in 2017, and that 5 million reduction in cost base that's caused by the consolidation of our Boston operations here to Cambridge. So one of the things we've been doing, just to finish off, is actually look at now, post the acquisitions, post the consolidations, how as we start moving into this year of delivering positive EBITDA, we will report data moving forward so that you can understand how the business is doing versus its objectives by measuring some tangible metrics. And we've talked to many of our top shareholders to gain input on this. So if we look at the group-wide revenue, you'd expect us to look at revenue growth, margin expansion, EBITDA improvement, customer expansion, customer satisfaction, and the amount of cash that we have. But for each of our business areas, remember we're trying to drive a products business that becomes the predominant part of the business over the next number of years. So we want to look at, yes, top line growth, but we want to break down revenue from catalog and custom models because they have different revenue profiles and profitability profiles. We want to report margin expansion, the number of cell line products, the number of reagent products because they're serving different markets, and the number of business-to-business relationships where we get that long-time annuity-driven revenue by our product being embedded into a third party's product. On the services, a little bit simpler. We want to see revenue growth, margin expansion, the depth of penetration we have in the top pharma customer base, and increasingly the improvement of our mean project size. That's clearly an indicator of whether we're getting stickiness, customer intimacy with our customers. And on the research biotech, we want to see the increase in the net asset value of our business on a DCF basis. So this is R&D payments and grants received, that's subsidized R&D, any milestone and annuity-driven revenue that we have, and the increase in our net asset value of our equity assets and spin-outs.